Today, we will delve into the fascinating world of cell biology and explore the intricate processes of photosynthesis. Photosynthesis is a fundamental process that allows plants, algae, and some bacteria to convert light energy into chemical energy, providing the basis for life on Earth. In this lecture, we will unravel the secrets of photosynthesis, from the structure and function of chloroplasts to the regulation of this vital process. So let's begin our journey into the world of photosynthesis. Chloroplasts, found in plant cells, use pigments like chlorophyll to capture light energy. This energy is converted into ATP and NADP, which are used to make glucose in the Calvin cycle. Chloroplasts have a double membrane and contain thylakoids, where photosynthesis occurs. Photosynthetic pigments, such as chlorophylls and carotenoids, are responsible for capturing light energy during photosynthesis. These pigments have specific absorption spectra, which determine the wavelengths of light they can absorb most effectively. Chlorophylls absorb light in the blue and red regions of the spectrum, while carotenoids absorb light in the blue and green regions. By having a variety of pigments with different absorption spectra, photosynthetic organisms can efficiently capture a wide range of light wavelengths, maximizing their energy harvesting capabilities. Photosynthetic organisms have evolved complex structures called photosynthetic light harvesting complexes to enhance light absorption. These complexes consist of multiple pigment protein complexes, which work together to capture and transfer energy to the reaction centers where the actual photochemistry takes place. The organization of these complexes allows for efficient energy transfer and minimizes the loss of captured energy. Moreover, the dynamics of these complexes enable them to adapt to changes in light conditions, ensuring optimal photosynthetic efficiency. Some photosynthetic organisms, such as cyanobacteria and certain types of algae, have evolved highly efficient photosynthetic mechanisms. These organisms utilize unique adaptations, such as specialized pigments and structural features, to maximize their energy capture and utilization. Studying these organisms has provided valuable insights into the fundamental principles of photosynthesis and has inspired the development of new technologies for harnessing solar energy. Photosynthesis involves two primary photosystems, photosystem I, PSI, and photosystem II, PSII. These photosystems work together to capture light energy and generate ATP and NADP. PSI mainly absorbs light with longer wavelengths, while SI absorbs light with shorter wavelengths. By functioning in a coordinated manner, these photosystems optimize energy transfer and ensure efficient electron transport during photosynthesis. Plastiquinone plays a crucial role in the electron transport chain of photosynthesis. This molecule shuttles electrons between SI and PSI, mediating the transfer of energy from the light reactions to the ATP synthesis process. Through its participation in the electron transport chain, plastiquinone contributes to the generation of a proton gradient across the thylakoid membrane, which is then harnessed by ATP synthase to produce ATP, a vital energy currency for cellular processes. While photosynthesis is a highly efficient process, a small fraction of the absorbed light energy can be converted into reactive oxygen species, ROS, during the electron transport chain. These ROS, such as singlet oxygen and superoxide radicals, can cause damage to cellular components if not promptly neutralized. Photosynthetic organisms have developed various mechanisms to prevent excessive ROS production and protect themselves from oxidative damage. These include the production of antioxidant enzymes and the synthesis of protective molecules. In addition to the conventional C3 photosynthesis, some plants have evolved alternate pathways, known as C4 and CAM photosynthesis, to enhance carbon fixation efficiency and reduce water loss. C4 plants, such as maize and sugarcane, spatially separate CO2 fixation and the Calvin cycle, enabling them to better adapt to high light and high temperature conditions. CAM plants, such as succulents and cacti, temporally separate these processes, fixing CO2 at night and minimizing water loss during the day. ATP synthase is a key enzyme involved in the final step of ATP synthesis during photosynthesis. This remarkable molecular machine transforms the proton gradient generated across the thylakoid membrane into the chemical energy stored in ATP. ATP synthase consists of two major components, the F0 complex embedded in the thylakoid membrane, which transports protons, and the F1 complex in the stroma, which catalyzes the synthesis of ATP. The rotation of the F0 complex drives the synthesis of ATP in the F1 complex, providing cells with a sustainable supply of energy. Excess light energy can be harmful to photosynthetic organisms, leading to the generation of ROS and potential damage to the photosynthetic machinery. To protect themselves, these organisms have evolved various photoprotection mechanisms. These mechanisms include non-photochemical quenching, which dissipates excess energy as heat, and the synthesis of specific pigments and proteins that can absorb and safely dissipate excess light energy. By utilizing these strategies, photosynthetic organisms can maintain their performance even under fluctuating light conditions. Photosynthetic organisms in certain environmental conditions, such as low CO2 availability, have developed carbon concentration mechanisms, CCMs, to enhance the efficiency of carbon fixation. CCMs enable these organisms to accumulate CO2 around the enzyme responsible for capturing it, reducing the energy cost and enhancing the overall efficiency of photosynthesis. Understanding the intricacies of CCMs holds promise for improving crop productivity and developing sustainable agricultural practices. Photosynthesis is a tightly regulated process that fine-tunes its activity in response to environmental cues. Various factors, such as light intensity, temperature, water availability, and nutrient availability, influence photosynthetic rates. Plants and algae have evolved intricate signaling networks that allow them to regulate photosynthesis and optimize energy capture and utilization. Understanding these regulatory mechanisms is crucial for developing strategies to enhance plant productivity and improve crop yields. In conclusion, photosynthesis is a crucial process that sustains life on Earth. Understanding the intricacies of chloroplasts and the regulation of photosynthesis not only sheds light on life's foundations but also offers potential improvements in agriculture and renewable energy.